Hello everyone, my name is Arij Bouazizi and I am going to present our paper, Knowing What to Label for Few-Shot Microscopy Image Cell Segmentation. In few-shot microscopy image cell segmentation, we usually train a deep neural network using a source dataset and then fine-tune it on the target dataset using a random support set. However, the random selection of support set may prohibit an effective fine-tuning performance. In this work, we propose a new approach on how to select and optimize the selection process of support sets from the target dataset. So our selection pipeline for pseudo-label segmentation learning includes a consistency-based scoring followed by a selection and fine-tuning process. So we propose our new self-supervised pretext task using the target dataset, assuming access to a collection of microscopy image datasets with a pairs of images and a binary cell segmentation labels. We rely on the source dataset to learn a generic binary cell segmentation function f theta, which is parameterized by the deep neural network with parameters theta. The target dataset contains images belonging to a different microscopy domain and a different cell segmentation task. So our main objective is to train a model to segment cells in microscopy images from the target dataset using a support set containing annotated images. So towards our objective, we consider a pool of unlabeled training images, T-pool, from which we select the samples of support sets based on a pixel-wise binary cross entropy function. The final model is then evaluated on the target dataset. We assume that the images belonging to the target dataset come from a different cell segmentation mask. Therefore, we seek to adapt the entire network, which means the encoder and decoder, to the task of cell segmentation learning using a pseudo-labeled dataset from the target dataset. To achieve this, we exploit the global contrast enhancement using histogram equalization followed by pixel-wise thresholding and a dilation filters to create pseudo-labeled target dataset. The model is trained by minimizing the average binary cross entropy loss on the pseudo-labeled dataset. The model is trained on pseudo-segmentation learning. We fix its parameters and start our selection process. Each microscopy image is augmented 3 times, each time with a different augmentation operation, namely auto-contrast, brightness, and sharpness. Then we accumulate the binary cross entropy score for each microscopy image between the prediction of the augmented images and the prediction of the non-augmented image. Finally, we select the images for the support set to fine-tune the final model. In our experiments, we use 5 microscopy image datasets and follow the same evaluation protocols as previous works. Instead of full resolution images, we make use of image patches to select the support set. We set the mean intersection of a union as our main evaluation metric and take the average over 10 runs. We compare our scoring function against Shannon entropy, MC dropout, and random selection. We show here the results of our scoring function in comparison to random selection. The red color corresponds to false positive, the green color to false negative, the black color to true negative, and the white color to true positive. Our method outperforms the random selection strategy in segmenting the images of 3 datasets. We further show on the right the mean intersection over union results average over all datasets. Our approach reaches the best mean intersection over union across all strategies. To conclude, we propose a support set selection approach for few-shot microscopy image cell segmentation. We reached better performance through random selection, Shannon entropy, as well as MC dropout. And in our ablation, we cover different components of our method. For more detailed results, please consider the main paper as well as our public code and models. Thanks for your attention.